It was a great time with Dr. GBS Raghava sir. And my dear participants, today it is a holiday, but despite the holiday, all of you are here for learning, and it's a great encouragement for us. So our second speaker, Dr. Kunal Roy, will be speaking, and he's also a great iconic academician, scientist in our field. He inspired in the field of QSAR, and the faculty members, those of who are teaching CAD, for us he's like a hero or icon because we are following his books. So to start this session, I like to request my colleague.
Thank you so much, sir. Okay, a very good morning to all. I, Sakshi Sharma, Assistant Professor, welcome all of you, all the dignitaries and faculty members on this AICT sponsored online Academy Faculty Development Program events, which is organized by Noida Institute of Engineering and Technology, Pharmacy Institute. I welcome Dr. Kunal Roy sir on this event. Dr. Kunal Roy sir is professor or ex-head of Department of Pharmaceutical Technology, JADAVPUR University, Kolkata, India. He has been a recipient of Commonwealth Academy Staff Fellowship, University of Manchester 2007 and Marie Curie International Incoming Fellowship, University of Manchester in 2013. The field of his research interest is qualitative structural activity relationship and predictive ecotoxicology. Dr. Roy has published more than 300 research articles in referred journals, Scopus Index 45 and total citation till date 10,557. He has also co-authored two QSAR related books with Academic Press and Springer Nature, edition 6 QSAR 10 chapter books. Dr. Roy is the co-editor-in-chief of Molecular Diversity in Springer and editor-in-chief of International Journals of Quantitative Structure Property Relationship. Professor Roy has been recipient of several awards including AICT Career Awards 2003 to 2004, DST Fast Track Scheme for Young Scientists 2005 and 2006 to 2009 awards from elsewhere. Bioorganic and Medical Chemistry Letters Most Cited Papers 2006 to 2009 awards from elsewhere. Professor Roy recently been placed in the list of top 2% science-wide authors database and the world ranked 81 in the subfield of medical and biomolecular chemistry. We welcome you, sir, and it's been a great honor to have you, sir.
Thank you for the nice introduction. Hope I'm audible.
Yes, Dr. Kunal, you are audible. Yeah, and your slide is also visible.
Thank you.
Yeah.
For inviting me to speak on this topic. And incidentally, Professor Majumdar has been my classmate during my UG and PhD studies. So it is a great pleasure for me to talk here. And my today's talk is on the topic of quantitative structure-activity relationships, basics of QSAR. And at the end, I would show one case study on mutagenicity dataset because this program is on cancer genomics, although I do not work much in the area of cancer therapeutics. QSAR is an acronym of quantitative structure activity relationship. This is a statistical model building approach. It correlates a response property, including biological activity, toxicity, or any other physiochemical property of chemicals using some numerical values, which are called descriptors. These numerical values or descriptors have a quantitative information about molecular structure and or property. And these descriptors can be derived either by physiochemical experiments or they can be computed using different levels of theories. And we can apply a variety of statistical tools to develop such models. So the basic notion of any QSAR is that the final biological activity is a function of either chemical structure and or their property. And as we are encoding the information within the structure in the form of numerical values, so we can write biological activity is a function of descriptors. If we see these three structures, chlorpromazine, promethazine, and ethopropazine, these three drugs belong to three different categories: antipsychotic, antihistaminic, and anti-Parkinsonian. But all of them share the same phenothiazine nucleus, only differing in their substitution or side chain pattern. So with changes in a substitution pattern or structural features, there will be changes in the biological activity, which is not only in the therapeutic category, but also in the type and magnitude of the interaction with the receptor site, their duration of action, and so on. So this kind of changes in biological activity with changes in structural features occurs in a very systematic way, and this can be actually mathematically modeled. So the objective of QSAR is to identify the pattern of changes in biological activity systematically with changes in structural features. And once the pattern is identified, we can apply this knowledge for prediction of biological activity of a compound or untested compound. QSAR can encompass both structure activity and structure property relations in a broad sense. It is an exercise of assembling, manipulating, and examining data obtained from physical, chemical, and biological, and also computational experiments, and correlating this to biological activity or property to be modeled using chemometric tools. In case our response endpoint is the property, then we can call this type of relation as QSPR. And in case this is a toxicity, then we can call it QSTR. But the basic notion is same, that the model response is a function of chemical structure and or property or descriptor. As QSAR is a ligand-based approach, so we have to start a series of compounds with their known structure and structural representation. And from their structure representation, we should gather information about their chemical structure in the numerical form. Or we can also depict their properties from physiochemical experiments, like say partition coefficient or acidic dissociation constant and so on. Or simply we can say, like say, number of carbon atoms or number of nitrogen atoms, number of double bonds, the aromaticity, etc. at the very low level as descriptors. Or you can go for some quantum chemical calculation also to get the high level information about molecules. But in a sense, we will have a column with the descriptor values, and there will be one, or in some cases more than one columns for the response or biological activity. And these descriptor columns may be numerous, and this response, which is important for our particular activity. Dimensionality and statistical method for QSAR model building. Now, this QSAR may be of two types. One is regression-based QSAR, and other is classification-based QSAR. In case our biological activity values that we will model are quantitative in nature, then we can go for regression-based technique. But in case our biological activity values are graded in nature, like say active and inactive, or toxic and non-toxic, so that also gives us some kind of information about the response or biological activity, but not very quantitatively, only in graded manner. In that case, we can go for classification-based QSAR building, provided our descriptor values are quantitative. And once we develop our model and we check the statistical quality and acceptability of the model using various statistical measures, and also we check the applicability of such model a new set of data through various techniques. And once we are satisfied with different metrics or measures, then we can apply this QSAR model for designing of new set of chemicals or prediction of the activity of untested chemicals. And models are also able to provide us with mechanistic interpretation of the biological activity being modeled. So we start with the molecular structure representation. We compute different molecular descriptions, appropriate statistical measures and fitting tools or prediction of different properties or toxicities or activities of compounds. So the flow of work will be like this. From molecular structure, we are getting QSAR table. We apply feature selection. We derive our model using appropriate modeling techniques. And finally, we are able to design some new molecules. Now, for biological QSAR, we have some requirements. Usually, the biological activity data should be taken in the log scale. And there are some requirements that the biological activity value should span over 4 to 5 log units to allow sufficient variation in the response values. And once we get our QSAR table, then we have to apply suitable data pre-treatment to remove noise from our descriptor table, because some of the columns may be intercorrelated or some of the columns may not be required for our particular response. So once you downsize our then we will go for division of the data set into training and test. So why this? Because we have our descriptor matrix and basically ill-defined, so we'll have some kind of, but we have to check the statistical quality and also the applicability of the model on the new set of data, and we have to check how good they are for prediction of new set of data. We have to go for this, it's called validation. Now, in many cases, we may not have additional data for validation purpose. So in those cases, what we do, we use our original data set into 2 parts. We divide into 2 parts. One is called training, that is used for model development. And another part is kept aside, which is called test set, and that is never used during the model building process. So this test set is actually unseen to the model development process. And once we develop our model, then we can apply this model for prediction of the unseen data, that is test set data. And as we already have the experimental biological activity values known for the test set data, we can then compare the model derived predictions to the experimental ones. And that gives us some idea how good or bad the model is for prediction on the new set of data. And in this process, we should think about a process called checking of applicability domain, because not all query compounds can be predicted using a given model. This is because QSAR is basically developed based on similarity principle. So similar compounds will behave in a similar manner. So if a query compound is sufficiently different from the training set compounds, and the model is derived from training compounds, so model gathers information and has been trained how to behave from the structural feature of the training compound. So if the query compound is sufficiently different from all the training compounds, it will not perform well for prediction of those query compounds. So you have to check for applicability domain, and we have to use different validation metrics, internal and external. I'll come about these metrics at a later stage. And when we are satisfied with the values of all these metrics, then we can apply this model for interpretation, for mechanistic explanation of different features, and we can also apply it for prediction of new set of data. Now, QSAR is not only applied for drug design and medicinal chemistry for modeling activity, admit properties, etc., and in the design and optimization of leads and refinement of existing drugs, but it is also used in material science for fine-tuning a property to get materials of enhanced operation selectivity and optimized process efficiency. For example, say optimizing of rubber, you want to get some polymer with optimum refractive index or some thermochemical properties, etc. So we can apply QSPR to modify their structural features, and we can get the new materials with optimized properties. We can also use QSAR for predictive toxicology, both including systemic toxicity and ecotoxicity, and they can be used for risk assessment, for hazard identification, hazard categorization, and prioritization of chemicals. In QSAR, we are using knowledge from experimental values of limited number of chemicals. And once we develop the model, we are able to use the model for prediction of a huge number of chemicals. And maybe these chemicals are never synthesized, or we do not have those chemicals physically, but we are able to predict their activity or property beforehand, before doing the experiment. And in that way, we can prioritize our experimental list of compounds, and we can reduce the number of compounds to be tested. For example, say if we have 10,000 compounds in our experimental line, and we do not need to synthesize and test all 10,000 compounds, but we can prioritize based on QSAR predictions that which compounds we will work with. And in that way, we can reduce our cost and manpower and time requirement. And instead of 10,000, maybe with 500 compounds, we can get one promising compound, which may give some useful results, which can help us in the drug discovery program, or program with some material development or in predictive toxicology. So QSAR is basically a filtering tool. So if anybody asks a question whether QSAR can give a drug or not, so for getting a drug, ultimately the synthesis and experimental testing is unavoidable. But QSAR can reduce the extent of experiment and experimental testing that one has to do. And in that way, it can reduce the time requirement, it can reduce the manpower, it can reduce the cost involvement, because drug development is a very costly affair. It takes a lot of time, say 12 to 15 years, and billions of dollars, and so much of manpower. So anything that can increase the probability of success will be helpful in this regard. And especially for admit prediction. As you know, many drugs are withdrawn even after marketing. So their admit behavior should be predicted at the very beginning, even before their synthesis. So the prediction of admit can be done very efficiently using various computational tools involving QSAR models. So similar thing applies also in materials and all predictive toxicology. So in the pharmaceutical science field, we are more interested for drug development. So I'm giving this slide which shows the development of new drugs starting from discovery and development phase with the targets profiling studies, toxicity studies, and in vivo studies in animals. Then comes investigational new drug application, followed by clinical research in phase 1, phase 2, phase 3. This takes a lot of time and resources. Then one can go for a new drug application, provided all data are satisfactory. And then if the drug is approved after marketing, also phase 4 clinical trial continues. It has been with clinical techniques like structure design, virtual screening, pharmacophore mapping, etc., has been able to candidates thrombosis, imatinib in cancer, raltegravir in AIDS, donepezil in Alzheimer's disease, hepatitis C, and so on. So now we cannot think any new drug coming without clinical experiment. So in silico are done very big drug discovery pipeline. And if a drug has to be drawn, it should be with a lot of money, because if we know that a drug has some problem at for recovery of the chemical, for chemical hazard assessment also, is very important. And recently I have become more interested in estimation of risk assessment of, especially from the viewpoint of ecotoxicology. So I apply QSAR mode for modeling drugs, for modeling the ecotoxic chemicals and modeling drugs. So the effect of hazardous chemicals and pollutants on ecosystems is a matter of great concern. And we have thousands of chemicals in the industries, and we are exposed to these chemicals through our everyday use product. But ecotoxicity data, experimental ecotoxicity data, are available only for a very limited number of chemicals, maybe less than 1% of compounds. So we need to fill the data gaps. And with the ever-increasing production of new chemicals and rising cost of experimental testing, and considering requirements to optimize resources, risk assessment of existing chemicals in use, various regulatory organizations like USEPA, OECD, and European Commission's Scientific Committee on Toxicity, Ecotoxicity, and Environment are recommending QSARs as an essential tool for ecotoxic risk assessment. We can also predict various physical properties and chemical properties and environmental and ecological and health effects of organic chemicals in the absence of experimental data. Such data can be used for regulatory purposes. As you know, the REACH regulation in the European Union that requires all toxicity and ecotoxic data to be supplied for chemicals, especially for production. In such cases, in case experimental data are not available, QSAR-derived data can be used for data gap filling. QSAR is very good for prediction of persistent bioaccumulative and toxic substances as an alternative to experimental testing. So QSAR is being used for regulatory purposes in regulatory settings, and this is encouraged by various regulatory agencies and also supported by REACH regulations. This is time and cost effective. It avoids animal experiment. It supports 3R principles, that is replacement, reduction, and refinement of animal experiments. And also it is ethically supported and supported by various regulatory bodies. So QSAR can be used for various endpoints in predictive toxicology, like hepatic toxicity, inhalation toxicity, genotoxicity, nephrotoxicity, development toxicity, carcinogenicity, eye irritation, skin irritation, etc. And in material science, there are several endpoints like carbon nanotubes, organic liquids, polymers, biomaterials, surfactants, and so on. So we again come back to QSAR model development, the statistical approach. This slide I have already explained. Only thing that if we get QSAR-derived predictions, and then if we plot the predictions versus experimental values, then we'll see a scatter plot, and if the model is good, the predicted value will be close to our experimental values. In that case, we'll see the low degree of scatter. So the points will be close to the 45-degree line. Now, if we see the evolution of QSAR, the concept started back in 19th century with Tom Brown and Fraser, who correlated the physiological action of strychnine with chemical constitution. Then in 1899, Mayer and Overton, narcotic potency was correlated with olive oil water partition coefficient. Then the first QSAR type relationship was applied mathematically for physical organic chemistry to correlate reaction rate with ionization constant of benzoic acid derivatives in 1935. And the true biological QSAR was done in 1962 by Hansch, who used the Hammett type of constant and applied it to hydrophobicity, getting hydrophobicity measure, hydrophobic substituent constant. At the same time, Wilson developed the mathematical model, and then came graph theoretical approaches, various connectivity indices, and there is still advancement in graph theoretical approaches after 1970s. In 1988, Kramer suggested 3D QSAR, comparative molecular field analysis, which is and after that, several other 3D QSAR formalisms have evolved. From 1990 onwards, ligand-based and structure-based approaches, virtual screen studies, and 1997 onwards, various higher dimensional QSAR also evolved. Now, the chemical structure representation can be done in formula-derived, 1D substructure fragment derived, 2D graph theory derived, 3D spatial geometry derived, conformation incorporated in 4D. In case of 5D, induced fit parameters derived. In case of 6D, 5D QSAR plus receptor information is also included. So nowadays, QSAR is actually used along with other structure and ligand-based approaches. Now, for QSAR, there are two major categories: regression-based and classification-based. And for regression-based model development, method of least squares and partial least squares, these two are very simple and preliminary ones that one can start with. In case of classification-based methods, discriminant analysis, logistic regression, etc. And nowadays, the machine learning is popular, and here classification-based methods like artificial neural network, regression-based, and support vector machine may be both regression-based and classification-based. There are some requirements for the endpoint data for biological QSAR. We have to use dose for fixed response type, like IC50, TC50, and so on. We should not use the dose required, that is the response for a particular dose. Like say, if you get some response, this kind of data are not suitable for QSAR. So we actually measure the concentration required to elicit a particular response. And this concentration should be taken in a molar scale. And obviously, there is an inverse relationship between the activity and concentration. So log 1 by C is actually important here. And there should be span of 4 to 5 log units. And there should be sufficient. So number of data points or compound should be higher, and number of descriptors used in the final model should be lower, so that we have good degrees of freedom. So a multiple linear regression model will look like this. So where in the left hand side we have Y, that is the dependent variable or biological activity, and in the right hand side we have X1, X2, X3. These are called descriptors. So these encode molecular structure information. So this can be like, say, number of some say hydrogen bonds, or some number of hydrogen bond donor, hydrogen bond acceptor, or number of double bonders, or it can be some physicochemical property also, like log P and pK. And A1, A2, A3, these are weightage of those regression coefficients. And with the mathematical or algebraic sign, they give us information whether a particular descriptor is contributing positively or negatively to the response, and what is the quantitative contribution from the quantitative values of A1, A2, A3. So during QSAR model development, this X1, X2, X3, these are known to us because they are directly obtained from structure or property experiment. And Y values are also known to us. So unknown values are A1, A2, A3, and also A0, the intercept. So these are obtained through a regression modeling approach. And once we develop the model, then we have to check the quality of the model obtained. Now, from the model, we can get the fitted values. So fitted values can be obtained from the previous equation. If we put the values of these descriptors in the model, and once we know the A1, A2, A3 value, we can calculate the Y values. So these are values, and then we can compare the calculated value with the observed value. So this is a comparison. Obviously, we aim at getting the low value of residual. That is difference between observed and calculated. But in a regression problem, some of the compounds will have positive, and some of the compounds will have negative residual values. So we cannot take sum of those. So we have to take square of those, and then we can sum those values. So sum of squared residual, this is our objective function, which should be minimum. Now there should be some reference point how we compare, or with what we can compare the sum of squared residual. And usually the mean of the training set response, that is the reference point, because no model. So y bar is equivalent to no model. So our model should be better than no model. So in the denominator, you can see we have compared the observed values with the y bar values, and we have taken in the similar manner sum of square differences. So obviously the numerator should be small and denominator should be high. So this ratio should be close to 0, and 1 minus this value should be close to 1. And this is called determination coefficient. And number of descriptors used. The same value of R or R square can be used using, say, number of descriptor 5 or number of descriptor 10 for the same number of compounds. And obviously, the model obtained from lower number of descriptors will be statistically more meaningful. And that's why instead of R square, one should use extraordinary, where we also use the information about the number of compounds N and number of descriptors P. So when one uses more number of descriptors, the value of R square will be penalized, or it will be lower. We have also variance ratio, which gives us information about the significance of the R square value and stability of variance. And denominator part, this is basically the sum of squared residuals divided by degree of freedom n minus p minus 1. And the value of F should be higher, and it should be significant at 99% level. And it has 2 degrees of freedom, p and n minus p minus 1, where n is the number of compounds and p is the number of descriptors. And standard error estimate is square root of sum of squared residual divided by n minus p minus 1. The value should be low. So these are the usual quality metric for a regression-based model, but this model, these metrics do not say anything about the predictive quality of the model. Now, in case of a classification problem, the metrics are different. As you know, in that case, the response is so we have toxic, non-toxic, or active or inactive. From that, we can get from predicted value, true positive, true negative, false positive, and false negative. And accordingly, we can calculate sensitivity, specificity, accuracy, precision, F measure, methods, correlation coefficient, and so on. Now, in March 2002, a meeting was held in OECD to formulate which modified to 5, and these are the 5-point guidelines of QSAR model development. Like there should be defined and defined applicability and robustness, activity, and mechanistic interpolation. So all QSAR models should be based on these 5 points. These are the according to OECD guidelines, starting with the OECD principle 5, OECD principle 1, that is defined in form. We cannot ambiguous algorithm in structure information descriptor generation in data pre-treatment in the application of measures, etc. There should be appropriate measures of quality like statistical quality metrics and validation metrics according to OECD principle 4. And also we should check the domain of applicability using OECD principle 3. And finally, according to OECD principle 5, there should be a mechanistic interpretation. We should take care about some points during QSAR model development, like data homogeneity that I have already mentioned. We cannot with different protocols. Inappropriate endpoint data should be avoided. It should be in the proper form for biological QSAR. We should avoid using collinear descriptors to avoid noise. Incomprehensible descriptors, some of the descriptors may be opting from some kind of matrix without having a physiochemical basis. So those kind of descriptors should be avoided. Error in descriptor values. If you are using some software that is unable to handle some kind of feature like some metal or some element like iodine, so that can give a wrong value of descriptors. So one should take care of those. Transferability of QSAR, the model that I generate should be transferable, reproducible by other group. Domain should be checked. Unacknowledged omission of data points should be avoided. Use of inadequate data. We cannot use 10, 15 compounds for QSAR model development. There should be at least some compounds, at least 30 to 50 compounds at the very low range. but more is the number of compounds, better is the statistical reliability. Narrow range of values should be avoided. Overfitting should be avoided by using filters. Lack of inadequate statistics is not preferred. Sometimes say particular molecular feature, say some keto-enol tautomerism is possible. So for some compounds, we have taken keto form, some compound for some similar compound, we have taken the enol form. So obviously the calculated descriptors will be different. The same nitro group can be represented in different ways, so computed descriptors may be different. So these are the things one should remember. Misinterpretation of statistics. In most of the cases, QSAR is being done by non-mathematicians, so misuse of statistics should be avoided. Consideration of distribution of residual important information about type of error present. Inadequate training test selection. So one should know how to divide the data set into training and test, preserving maximum diversity. Failure to validate QSAR, QSPR properly, and mechanism. So each of these points were explanation actually. I can. So validation is the applicability of the model on the new set of data, because when a model is developed, it is not used for calculation of the endpoint or response for the training data. It will be used for new set of data or unseen data. So we need to know how good or bad the model is for new set of data. So for that, we have to go for method for validation. And this is in general of two types, internal and external. In case of internal validation, we can use the same set of data that we have used for model building. So what we can do, from the training set of compounds, we can remove a particular percentage of compounds, and using the remaining compounds, that is reduced data set, we can redevelop the model. And we can then predict the omitted compounds using this particular model obtained from reduced set of data. And we can get the predicted values for the omitted compounds, and this we can repeat. So this repetition can be done either using a leave one out approach, where we can omit only one compound in each cycle, or we can do leave many out, where a percentage of compounds, say 5% or 10% of the compounds, are removed in each cycle. And finally, we will get the predicted values for all the compounds, and we can apply the metric which is called Q square or cross-validated R square, which looks like similar to R square, simple R square. Only thing is that we are using here predicted values instead of the fitted values. In case of external validation, we should have a separate set of compound. Here we do not use training compounds. We will use either test compounds or external compounds, and we will apply the model for prediction of the new set of data, and then we'll apply the similar kind of metric, R square pred, which looks like similar expression. Only difference is that we are using here the predicted value of the test compounds, and all the values of here for reference the Y bar or mean value of the response of the training set compounds in R square pred, or these also called Q square external F1. There are other variants also like Q square external F2, F3, and so on. There are some other methods like bootstrapping, which is essentially an internal validation technique where one can do sampling, and also Y randomization, where one can shuffle the Y values, keeping X matrix intact and redeveloping the model from the randomized Y column. And obviously the quality of the random models will be here. And these are explained more here, but go into this detail because we have limited time. As I mentioned, that we have to select the training compounds very judiciously, so that we place the most diverse compound in the training set. And also we have to keep sufficient number of compounds in the test set. We should not keep 3, 4, 5 compounds in the test set, because that doesn't give proper justification of checking of prediction model. There are different ways of selection of training compounds based on similarity principle, statistical design, coherence, self-organizing map, K means clustering, K stone, spear exclusion. So, but also we can use a method that is called activity ranking, and also one can go for random method of division of training and test. But in that case, one should do it several times. And finally, applicability domain. I have mentioned that applicability domain is very important for prediction of set of data, and there are one can go for leverage, which is often hat matrix. Hat matrix is basically this one, the diagonal element that gives a distance of between the x values of ith observation and of all x. And the calculated value should be in some threshold value, that is threshold is 3n, where is the number of variables plus 1 and n is the number of compounds. So there is a great degree of debate between internal and external validation, but we will not go into that. We prefer both. We have to satisfy both the metrics for internal validation and also external validation. However, cross-validation has been criticized by some group of authors, and external validation has been considered as the gold standard for the quality of prediction for the set of data. So one must go for external validation of any developed model. Okay, so now I have mentioned here the basic methods of validation, and briefly about some metrics for judging the quality of the model, both in terms of statistical stability and also their predictability. And we have developed different tools for model development, mainly for multiple linear regression and partial least squares model development, and also some classification tools. And we have several tools for validation purpose, domain using standardization approach, and also indicator and intelligence, that is when we have models for using model, making predictions from multiple models. All are available in these two sites. So these two sites host a range of different QSAR tools that we have developed in our laboratory, and all these tools are available free of cost. One can download, and these are Java-based tools, so one should install Java before running these tools. And one can go for the QSAR model development and validation. For descriptor calculation, one can use other tools like the alpha descriptors, Dragon, panel descriptors, and so on. And from the descriptor values, using descriptor values, one can give input in the Excel file, and the output also one will receive using Excel file option. Now I'll show one case study of application of QSAR in identification of mutagenicity mechanism of nitro and amino aromatic compounds against Salmonella typhimurium species. As you know that organic chemicals are very important for us because we use organic chemicals in various spheres of our life, in dyes, in polymers, in pesticides, in textiles, in pharmaceuticals, in explosives, food substances, and so on. So we are surrounded by organic chemicals. We are exposed to organic chemicals, but not organic chemicals. Not all organic chemicals are good for us. Some of them are toxic, they may be mutagenic also. So mutagenic compounds are different from carcinogenic compounds in that they do mutation before causing carcinogenicity. And this mutation can occur through a variety of mechanisms like alkylation, oxidation, amination, and hydrolysis of bases. And there are some additional like photocatalytic and so on, and coordination. So usually for mutagenicity test, MS test is applied where histidine-dependent Salmonella is used, and histidine lacking media is used for this test. And for nitroaromatic compounds, TA98 strain of Salmonella typhimurium is used, which has a full complement of nitroreductase enzyme required to activate the reduction reaction of nitroaromatics, whereas aromatic and heteroaromatic amines demand the presence of exogenous metabolic activation system, that is S9, to initiate oxidation reaction. Now we have used here a data set where we have this that is TA98 strain plus S9 and TA98 strain minus S9, that is in presence and in absence of the oxidative enzyme. The data are available, and we have used this data set for our model development purpose to identify the features required for mutagenicity using this particular data set. And as I've already mentioned, that is a ligand-based approach, so we need to present the structures appropriately through appropriate structure presentation, and then we have to go for descriptor calculation. So here we have used only two-dimensional graph theoretical representation of molecular structure, because in case of 3D, one has to go for conformational analysis and energy minimization. That some problem producing and the 2D descriptors, once calculated, they will always give the same values, which is not the case in case of 3D. You can get in different range, different descriptive values. And that the 2D models are used, and one can start quickly, which is very good for the initial idea about the screening. We have calculated descriptor, then we have applied the data pretreatment to remove the redundant variables, to reduce noise, etc. And then we have gone for data set division to training and test. And with the training set, we have developed our model, and then test set used for prediction. Usually we use different data set division methods like Euclidean distance-based method or Kennard-Stone, or sorted response-based method. And we show the best model that is opting for a particular division method that is giving the best results. Now, here in this particular case, here I've not gone for details of the feature selection and the descriptor selection. Usually, we can go for voice-based method or genetic algorithm-based method to identify the descriptors which are important for us. And also sometimes we use multi-layered approach for descriptor selection. That means we apply stepwise method and identify some of the descriptors. We omit those descriptors from the descriptor matrix, and then apply again stepwise regression to identify a new set of descriptors. In that way, we can make descriptors. After that, we apply a base subset selection or genetic method to identify the descriptors that are important. Derive a number of models. So one of the model for the TA98 minus S9 is this one. As you can see, in the left-hand side of the equal to sign, this is actually Y, the mutagenicity value in the appropriate form. In the right-hand side, you see some numerical values and names. So, for example, this is a connectivity index type of descriptors with the coefficient 4.210. This means that this descriptor has a positive contribution. With the increase of the value of the descriptor, it will increase the mutagenicity value. And sometimes we see the negative coefficient. Negative coefficient means when we increase the value of this descriptor, so the number of circuits here, this will actually reduce the endpoint value. So in this way, we identify the involvement of different descriptors. So this is another model for the endpoint. That is TA98 minus S9, that is without S9. And so once we develop the model, we are able to calculate the endpoint value for both training and test compound. Then we plot it. You can see this is a good degree of good scatter plot, because these are close to the 45-degree angle line. So that is the fitted values for training compounds, and predicted value of the test compounds are satisfying with the observed values. Now, regarding mechanistic interpretation, so if we try to interpret the different descriptors here, which I'll not go into details here, we can identify different features like, say, B09 is between two nitrogen atoms, or number of circuits. So that gives us the number of, say, polycyclic aromatic rings, etc. The distance between two Cl, that is Cl is lipophilic, so, and that is present in a moiety where that is the minimum topology, the topological distance of 8 is possible. So that means there is sufficient bulk. So this information gives us whether interpretation. For the other endpoint, T98 plus S9, in presence of S9 enzyme, we have this model And along with the model, we have to also see the quality of the model. Here, R square is 0.701, and Q square, leave one out, Q square is 0.680, and R square paid is 0.696. So usually this Q square and R square paid value should be more than 0.6, and R square value should be more than 0.7. So usually in biological QSAR, we do not get a model with R square value very high, like 0.9, etc. It is not possible for all data sets, especially where there is some structural diversity. In generic series, it is to get 0.9, but in case of diverse type of compound, the 0.9 are difficult. And I would also add here, we do not stress on R square value, on Q square, that leave one out and leave many out Q square values, and R square paid values are more decisive indicators. And we also stress on error-based metrics like mean absolute error or RMSE values as an indicator of the quality of the model. This is the second model for the TA98 plus S9 models, and these are scatter plots. And these are some of the molecular features enhancing or reducing toxic mutagenicity of TA98 plus S9, showing different kind of features like number of imidazoles, number of pyridines, distance between two nitrogen atoms, etc. And also we have used some fragment-based descriptors, same descriptor, to identify different features in the molecules present, to identify the fragments which are important for going, but want to see what is the study from a layman's point of view. We'll see that we have identified 4 features here: the lipophilicity, effect of lipophilicity on mutagenicity, effect of electronegative atoms, effect of unsaturation, and effect of branching. These are the 4 features. Now, regarding lipophilicity, there are many polycyclic aromatic compounds, planar compounds, big molecular size are present, and these are lipophilic, so this increase membrane fluidization, and they can be accumulated, and then there will be metabolic activation, and followed by free radical generation, which can lead to mutagenic effect. And also presence of electronegative atoms can enhance the that is nucleophilic attack, and it can lead to intercalation and subsequent alteration in DNA. Effect of unsaturation also increases and addition of liquidity, followed by metabolism and reactive oxygen species generation, and various adduct formation, adduct with adenine and guanine bases. And effect of branching. Branching actually has opposite. So it has opposite effect. Now, the models that we have generated that have been incorporated in the ALVA runner, that is obtained from ALVA Science. So ALVA runner is an expert system that can help one to use the expert system to predict some endpoint. Here, the mutagenicity endpoint can be predicted using ALVA runner. So this is a third-party software, and from their website, one can use the expert system for prediction of some query compounds for their mutagenicity potential, where our models have been used. And this wants to know more about QSAR from beginner's point of view, one can go through book Understanding Basics of QSAR for application in pharmaceutical science and risk assessment, published by academic press. And in more brief form, one can the slides are changing slowly, actually. I don't know why. So the slide hasn't come, I think. And this is one. The previous one was this one. It's not showing the previous one anyway. This is brief form about QSAR. So the novice user can use this book for knowing different aspects of QSAR, book of application of QSAR in drug design, predictive toxicology, and risk assessment. And the more advanced application of QSAR in different uses like pharmaceutical, chemical, food, agriculture, and environmental sciences. And more specifically, the application of QSAR and related techniques for computational modeling of Alzheimer's disease, modeling of drugs against Alzheimer's disease, multi-target drug design, and for drug repurposing and for ecotoxicology QSAR. And more recently, the in silico modeling of drugs against coronaviruses. And forthcoming is chemometrics and cheminformatics in aquatic toxicology. So these books we have contributed from our laboratory. Thank you very much. So if the person have any question, I would be happy to answer.
Thank you so much, sir, for such a motivating lecture. It's really very knowledgeable and informative data for all the faculty members. So there is one question from Abhijit Debanan, sir. He is asking that how do you differentiate between QSAR and machine learning?
So machine learning, as you know, in case of machine learning, we apply basically regression and classification based methods. Like say, artificial neural network is a regression based technique, whereas support vector machine can be used for both regression and classification. But the basic difference is that the QSAR is basically a statistical approach where we have to use different statistical degrees of freedom for reliability, etc. That means we should use more number of data points and limited number of descriptors in the model so that we can get statistically reliable model. But in case of machine learning, number of although but there are different means to avoid those kind of overfitting, which is usually absent in case of QSAR. So QSAR is basically used using the classical theories of statistics, whereas in case of machine learning, we can use more number of data and more number of descriptors simultaneously for prediction purpose. And usable prediction than interpretation. Whereas in case of QSAR, as these are more transparent because you can see the model and you can interpret different descriptors and their meanings. So the statistical model building is preferred for mechanistic interpretation of some response or biological activity or toxicity, whereas machine learning models are better for prediction purpose. So there is a, I would say, compromise between the interpretability and predictability. So models may be somewhat less predictive, but about physicochemical, but machine learning models are more predictive, but less. They give less interpretation about the physicochemical basis.
Okay, thank you so much, sir, for such an informative knowledge, and we are very grateful to you for having a part of for these sessions and giving your valuable time. Now I invite our convener, Dr. Rupa Majumdar ma'am, to address the audience.
Dr. Roy for delivering a very informative lecture on quantitative structure-activity relationship, the basics, a mutagenicity dataset case study that you have explained from the very root level, which will be very much helpful to most of the participants who have attended your lecture today. I am really feeling proud to have you within us today as an eminent researcher and researcher of the day and the topmost, one of the topmost scientists of the world and an expert speaker of the today's event. My pride is also due to the fact that we are very good friends since 1989. That is approximately for the last 32 years starting from our JU days. Dr. Roy was a topper of our class, gold medalist of both the BPharm and MPharm courses, and had shown his sincerity and brilliance throughout his student's life. Dr. Roy was very much systematic throughout and always focused on whatever assignment he had to perform. His intelligence, along with all these qualities in him, have made him an excellent researcher and an eminent scientist and expert with so many followers in the field of QSAR. So I really feel proud when I hear from someone that I am a fan of Dr. Kunal Roy. So really great. And we would like to have you within us again and again in our future events also. Thank you, Dr. Roy. Thank you so much. And thank you for spending your valuable time with us today.
Thank you very much, Upa. I'll be glad to be with your future program also.
Of course.